Yeah, it looks like we've got quite a few people who have tuned in this afternoon. So I think we'll get going. Tēnā koutou katoa, tēnei te mihi kia koutou. Thank you for joining us this Friday afternoon. And welcome to the first in what we hope is a series of presentations on projects that have been supported by Genomics Aotearoa's Indigenous Genomics platform. I'm just going to hand over to Aden, who's one of the project team members, and he's going to open us with a karakia.
Kia ora everyone. I'll just get us started with a karakia, sort of get us all in that frame of mind of what we're doing today. And hope this is a good wānanga this afternoon. Matua tama, wairua tapu, me ngā anahira pono, me te maanga he tautoko mai, ānei, āke nei, āe. E te maanga i ngā anahira pono me te tokotoru tapu, kia tatu mai anō koutou ki wānui tēnei hakamoemiti o mātou, kei tātari ngā kupu katoa, e haka puaki nātu ki roto ki tō koutou kororiatanga. Ko koutou mā te timatanga me te hakamutunga mai o mātou tumanako nei I roto I tō koutou kororiatanga. Ko koe te maanga e hei tautoko mai, ānei, āke nei, āe. Kia ora everyone.
Ngā mihi kia koe, Aden. Yes, so welcome again, everyone. So for those of you that don't know, our GA Indigenous Genomics platform focuses on supporting projects that are led or co-led by mana whenua. So the projects address issues that have been identified by mana whenua themselves and deliver direct benefits to the mana whenua involved. So today we're going to hear from hopefully the whole project team that have joined us. That's Debbie Martin from Te Rarawa, Michelle Knapp from Otago, our Kauri Dieback Rangers, Takapari Murray, Aden Moses and Hama. My apologies, Hama, I'm not sure of your surname. And Amanda Black from Bioprotection Aotearoa and Lincoln. Oh, and of course, our very own Peter Dearden. So first of all, we're going to have Debbie will give an overview of the Kauri Dieback iwi collaboration program, and then that will be followed by a presentation from Michelle on the project itself, and then we will move into a discussion with the team. Feel free, for those of you in the audience, if you have pātai or questions that come up, to pop them into the chat, and we'll hopefully get a chance to address all of those during our discussion kōrero. So with that, I will hand over to you, Debbie.
Ngā mihi. Just going to share my screen. Okay, right. And okay, tēnā koutou katoa, ko Debbie Martin ahau. I am the program manager for Kauri Dieback, the iwi CoLab. So I thought I'd explain a little bit about the iwi CoLab because it's a little bit broader than just the Te Rarawa ranges. So Kauri Dieback, who are we? We're a unique collaboration of 4 te tai tokero iwi, Te Rarawa, Te Rarawa, Ngāti Kuri and Ngāti Wai. We have a shared vision to ensure our coding ngāhere thrive for future generations. In late 2020, with encouragement from MPI and DOC, the 4 iwi came together and the iwi CoLab for Kauri Action, or Kauri Dieback, was formed, and an application to DOC Jobs for Nature was compiled, and that activated Kauri Dieback from May 2021 to May 2023 under that Jobs for Nature funding. And so during that time, all 4 iwi stood up their own team of rangers who were then trained to use a variety of tools in the mission to protect and maintain our taonga species, Kauri. We also had a project team under Jobs for Nature, but under our new funders, Te Kina Kauri, we have less money, so I'm the project team. Our rangers are trained to do surveillance and monitoring, to use mātauranga Māori and rongoa in treatments, and they also do education and awareness programs. So the Kauri Ora iwi CoLab offers an opportunity for our iwi to work together to increase the capability and capacity of our people to work on their whenua for the benefit of Kauri forest health. At the same time, even though we mahi tahi the four iwi, each iwi maintains its own mana motahake within the hapu and whānau context of Te Rarawa, Te Rarawa, Ngāti Wai and Ngāti Kuri. As I had mentioned earlier, with the government establishment of Te Kina Kauri, or the Kauri Protection Agency, the iwi CoLab were able to secure future funding once our Jobs for Nature funding had ceased, and we are able to continue our mahi through to mid-2026. Our vision is quite clear. E kore te kauri e tū moki moki, our kauri never stand alone. So what that kind of encompasses is that the forest ecosystem stands with kauri, as do all hapu, iwi community and government. The protection of kauri and the enhancement of their mauri in turn enhances the collective well-being of those who stand with them. So our vision is inclusive in the broader sense. So this map shows the spread of the rohe of the four iwi CoLab in Te Tai Tokerau. We are the access to the whenua through our hapu, whānau and marae relationships. The forests included are significant. They cover around 50% of the Kauri forest in our region and include the two most significant old growth forests in Northland. That's Waipawa under the care of Te Rarawa, and the Warawara, which is under the care of Te Rarawa. Our rohe also covers two of the most significant destinations for international and domestic tourists, Tāne Mahuta down in Te Rarawa lands, and also Te Rerenga Wairua up in Ngāti Kuri. So these next couple of slides just cover a little bit of the mahi that our rangers undertake. Some of the types of work they do are surveillance and monitoring that includes using GIS, data management. We undertake Kauri operations, tree health assessments, diagnostics, soil sampling and treatments. Also broader tile forest operations, so we protect, control pests, plants and animals, and also assess stream health. In the biodiversity protection, I've mentioned trapping. We also do fencing, look at fire breaks. We also harvest, propagate and plant kauri. We have some brilliant science and research participation, such as what we're here for today. We do community outreach, including working as ambassadors on tracks to educate the public as to hygiene protocols, and also tell them a little bit of the history of the area that they're walking into. Yeah, I think one important aspect of our mahi is when we have whales strand and become deceased within our rohe, we undertake what's known as flensing. where the whale is given a name, and then the resources are taken and used. We also use some of the whale remains in our rongoa treatments, which we are trialling with sick trees. So that's my kōrero. Just a brief introduction to Kauri Dieback Iwi CoLab. So I'm sure no one's got any pārtai, but if you have, you're welcome.
I just thought, Debbie, we possibly have some international people that have zoomed in who may not realise the significance of Kauri. So just very quickly, our New Zealand Kauri trees are endemic. They are considered taonga or treasures by Māori, particularly by those tribes that live with Kauri in their areas, but they're also nationally appreciated as an iconic species. And that's what this project is all about, caring for Kauri. What are the main challenges you are facing alongside funding? Acknowledging that funding is always an issue.
So many challenges, it's hard to it's hard to define what the main one is. Yeah, funding is an issue. Yeah, as I said, we're only funded up to mid next year, and we don't know if there is a budget for mana whenua to do the type of work that we do. Going beyond that, we should know from central government at the end of the year. I suppose a big challenge is also working with government, to be honest, because you know they have we have a national pest management plan which is related to Kauri. So Kauri have a not all of them, but there's a disease that exists which Michelle, I'm sure, we'll talk about a bit later. So that's why the National Pest Management Plan's being set up. So we have to work within its confines, and we also have to work with the tools that the government provides. So they're kind of the surveys we undertake and the GIS mapping system, which is run by LINS. So there's been a lot of teething problems with our tools. Which we're not actually in control of. So it can quite often stymie the ability for our rangers to get out into the field and do what they need to do. The weather is also a challenge. You can't go into the forests when it's wet. So because that helps the spread of the pathogen. So we sort of have a dry season and a wet season in the north. So over the summer months, that's when our rangers are out in the bush sampling and monitoring. And then in the sort of winter wet months, that's when their education and engagement work stream kicks in.
Thanks, Debbie. Michelle, we'll pass over to you now.
Yeah, so thanks, Debbie, for the introduction of the whole project. And obviously, this is quite a wide-ranging work. And what I'm going to talk about is just a very small aspect of it. Debbie has already hinted at it. One of the major challenges that the Aotearoa team is dealing with is Kauri Dieback, this pathogen that is essentially 100% lethal once the tree is infected, and that is very challenging to manage. And the Aotearoa team had identified that essentially detecting it quickly and ideally in the field on the spot would be a major help with managing the pathogen. And so this was a project funded by Genomics Aotearoa. I came into this relatively late, in late 2023. Specifically, I was brought in by Amanda Black, specifically asking whether I might be able to help with developing a tool that allows for Kauri Dieback detection in the field, and that can be used by the community where, samples don't have to be sent away. I also noticed that in shuffling around the author list down there, I misspelled my own name, so this is starting really well. In any case, so most of you will be familiar with Kauri Dieback. Just a very quick recap. Kauri Dieback is caused by this water mold, this fungus-like organism, Phytophthora agathidicida. So it's the same genus as, for example, the pathogen that causes the potato fly, which was one of the key factors in the Irish famine. It's got a relatively complicated life cycle, so it lives in the roots mainly of the tree. It doesn't go very far up the trunk. You can find it in these mycelia, so again, very fungus-like. And it produces oospores. These are little units, very hard, that sit on the outside of the roots, that also are in the soil around the roots. And they kind of protect the organism in times when the environment isn't particularly good. Then under good circumstances, which for Kauri Dieback is essentially wet and cold, they start releasing these zoospores, these things here, and they have tails. They can actually move around, and they move around the soil and find a new host and infect it, and then the whole cycle starts again. Because this is such a big problem, and because it really affects kauri forests, and increasingly so, it is monitored, and there's also surveillance tools available to detect where it's present and not. The standard for this is usually this technique here, which is called soil baiting. So essentially, the Aotearoa team goes out, collects soil samples from around the trees that they want to test. Then these samples get sent to specialist companies. Here our collaborators Biosense, for example. They incubate the soils with water, put some organic matter on top. That could be lupin seeds, or it could be just pine needles. And these then attract the zoospores from Phytophthora. They accumulate around this organic material, and then you can use the organic material to detect if Phytophthora is there. Problem is, of course, for that, samples have to be sent away, raising all sorts of questions for sample sovereignty, data sovereignty, but also that process takes quite long. It can absolutely take more than a month from taking the sample to knowing whether there is any Kauri Dieback in there. And that, of course, is a delay that you don't want to have in a pathogen that is so infectious and that can be spread quite easily. With the soil baiting, I think there's about 60% or so detection rate if you sample around trees that do have symptoms. But then again, you're mostly interested in those that don't have symptoms yet. So that drops to about 20% detection rate there. Because it's such a faff to do this test, it is quite expensive and can be done only by very specially trained personnel in a few places. And that lag time really has an impact on decision making, obviously. So if we could develop a tool that can be done on the site, close to the sampling, and it is quick, that immediately provides the result, that would be a major step forward. And so that was one of the challenges that we tried to address last year. And by me, that is very much, by we, that is very much a collaboration. So we've got Hama, Takapari, and Aden, who are here. Debbie leading the site at the Rarawa, and we have Amanda Black and myself working in the lab. There's also a team at Biosense. So it's quite a large collaboration working here to figure this out. When we started, basically, the original plan was this. So first of all, because we only had funding for about 11 months, we were on a tight timeline here. So it was clear from the beginning that what we needed to do was, we couldn't develop any new tools or anything. We needed to figure out a way where we can just cobble different tools together that are already existent, that are already working, and make them work for us to detect Kauri Dieback, ideally in the field, ideally simply and ideally on the side. Because all the testing previously had been done with soil samples, that is what we started out with. So we wanted to target soil samples, extract DNA from these soil samples using environmental DNA technology. And from this environmental DNA, we would then amplify, target Phytophthora DNA to see if Phytophthora was in the extract or not. And in principle, that's also fine. This is also not in any way unprecedented or new. Essentially, you can set up a very small lab that you can take into the field. That was the aim. So for example, one of my former PhD students, Stefan Prost, has done this, even taking a lab like this into the jungle in Ecuador, doing some biodiversity monitoring there. Joe Stanton has done similar works, and Joe's been part of the team in the first half of the project. Has done similar work in Africa, looking at viruses in field plants. So again, this was something we should be able to use and put together. And so with some advice from Stefan and a lot of development from Joe, we put a lab together that is essentially fits into a backpack, can be taken out to the site, can be used to extract DNA in the forest. And then the idea was we use that to detect Kauri Dieback. In terms of life cycle, it meant obviously we weren't quite sure what to start with and what to target here. And the first part of the project, I was partially overseas, and Joe did a lot of this work. And she tried to figure out essentially how best to isolate Phytophthora and Phytophthora DNA from soil samples, from environmental samples. Again, we drew on methodology that had already been developed. The team from Wellington has been working with these sort of methods, where essentially you pack soil into tea bags. The relevance of the tea bags is that their pore size keeps the soil together, but the oospores are small enough that they can get out of the tea bags. You then put the tea bags in a syringe. You flood the syringe with water. You put a filter that is tight enough to collect the Phytophthora oospores at the bottom, and you press the water through, and then Phytophthora oospores accumulate on the filter, and you extract DNA from that filter. Joe did a lot of work in this space, and she did a lot of testing. She also went to the lab and tried to attract zoospores specifically, use different substances, and you can actually bait them with sort of, I think it's ethanol or something in your liquid, and they all come around. So there's been a lot of trying, and by the time we were finished with this, it was about May, and we had our first meeting virtually at the time with the team up north. I wasn't there at the time, so Joe presented, and the feedback was, ideally, for this to work reliably, you should work with soil samples of at least 300 grams or so, which in a DNA extraction context, you can't do in the field. You need massive centrifuges that nobody can carry around. And if you want to work in the field, you're limited to what you see on these pictures here. It's about 10 grams. So after a lot of fuffing, we just didn't think soil was the perfect way to go. But what also came out of that first meeting was that a lot of the samples taken contain a lot of root material, and they deliberately contain a lot of root material, because that's also where you would find Phytophthora and Phytophthora oospores. And so we were wondering whether it might actually be better to, instead of puffing around with lots and lots of soil, we just take the roots out of the soil, because the mycelia of infected trees, mycelia from the Phytophthora organism should be in the roots, so they should already be accumulated in there. You should also have oospores on the outside of the roots. So this might almost be kind of a natural accumulator of the pathogen, and might therefore be more suitable or more easy to use in the field. And so we started building our protocol on this. Again, this was end of May, and I was starting to be worried that on our first meeting in person up north, where I was going to meet with Takapari, Hammer, and Aden to present an approach that we could use, I was starting to worry that I wouldn't have anything to show for. But it started working once we started going to the roots. So the key thing is, roots are, of course, just plant material. So to extract DNA from these roots, you don't need anything special. You can really just use your QIAGEN plant extraction kit. A small problem is that in the field, you can't crush your roots under liquid nitrogen, because you don't have a big container of liquid nitrogen with you. So we had to crush them fresh in our 1.5 milliliter Eppendorf tubes using these jerry-rigged number 8 wire pestles, also known as pipette tips, that have the tips burned off. And to test this, I didn't have any samples from up north yet, so I went to a nursery and got Karl the Kauri. Karl the Kauri is a young kauri tree, and the roots from Karl the Kauri was what I used down here to just test the protocol, to develop the protocol and see how it works. That turned out to be a bit misleading, but more on that in a bit. So this is essentially the protocol that we took up north on our first the first trip up north to present the protocol and discuss with the team to see if that was in any way suitable. So you take a root sample, crush that in your little mortar and pestle that you make up. You extract the DNA using that mini laboratory, which you can set up anywhere in the field, in your kitchen, anywhere. It's very small. It doesn't include a lot of material. And then to detect Phytophthora, we thought the best way to do that at that point was using quantitative PCR. So there is a machine called the Ubiquitome Liberty 16, which is this here, which is a portable quantitative PCR machine. It's got its own battery, so you can run it in the field, and it also connects to your cell phone. So basically, what it does is you put DNA in there and PCR primers, and then you help, essentially, if your target DNA is in there, so if Phytophthora is in there and you're targeting Phytophthora, then you will see at some stage an amplification plot like that. Okay? Now, we figured that the best way to do this, to make sure that everything we're doing actually works, is to have in every reaction a positive control, which we use Kauri, and Phytophthora as our target, which means that we use the same DNA across all reactions, but in half our reactions, represented here by our top line here, we use Kauri primers, and in the other half, this bottom line here, we use Phytophthora primers. Primers have been optimized to work at exactly the same temperature, Kauri and Phytophthora, so we can use it in one reaction. And so the outcome, ideally, is what you see here. All the Kauri, or well, except for that one, but all the other Kauri extracts amplify. You can see the number of cycles at which they amplify. The lower the number, the higher the concentration. And then, because this was a sample from Karl the Kauri, and Karl the Kauri is not infected with PA, you see all the PA amplification does not show anything. So you've got flat lines for all your PA, and you've got amplification for all or nearly all of your Kauri, which means your extraction has worked fine, your amplification is working fine, and there just isn't any PA in this extract. Here's an example of how a positive detection would look in this system. So I've just picked out one to make it a bit easier to spot. Here's again our Kauri in the top line, and I picked out one sample here, and PA Phytophthora in our bottom line. So you can see Kauri comes up first because there will always be more DNA from the host, from the Kauri, than from the pathogen. And then usually 4 or 5 cycles later, you see Phytophthora amplifying, and this is kind of a good example of positive detection. Here is Phytophthora in your sample. So that all sounded great in theory, and well, went up and Takapari, Hammer, and Aden had a go at it and tried it out. And we discussed how that would work in the field. And it became relatively quickly clear that there was a major problem here. In fact, there were 2 major problems. The second one I found later, but the first major problem was that it just took way too long. On a subsequent trip, the team actually took us out to do some sampling in a kauri forest, highly infected kauri forest. And on that trip, I had one job, and that was writing down the numbers on the bags. And the team was throwing the bags so quickly at me that I couldn't even keep up with the numbers. So they're working extremely fast, extremely efficiently, and they have to work that fast and efficient because there's a huge area to cover. And the thought of then at every tree site having to set up your lab and spend an hour or 2 or whatever DNA extracting just completely interrupts it and actually makes covering the area not possible. So the process takes too long for in the field. It's theoretically doable in the field, but it takes too long. There are ways to speed this up, enzymatic extraction, for example, but the only machine that was suitable for that at the time is currently no longer on the market. It's the PDQX. So with the tools we had, we just couldn't make it work in the field. Now, that problem, after some discussion at the Airbnb in the evening, we figured we'd be able to solve reasonably quickly. And that's basically by setting up a kitchen lab. So the idea to make it work would be, for example, that the team splits up. One part of the team does the collection, provides the samples to the other person who stays at home and does all the extractions. So that basically with 1 day delay, all the samples are extracted, and you have 1 day after sampling, you have a detection or you don't have a detection. So that's a significant speed up of the process, and it can be done on site. Samples don't have to be sent away. This could work really well. Well, it could work really well until the boss starts distracting the team, but we'll gloss over that. So that was a logistical problem, but there was also one that was a bit more of a worry, and that was that at this time where we did get PA detection, it was too spotty, too sporadic, and there were way too many false negatives. So false negatives are simply when your Kauri doesn't amplify either. And then you know something's wrong because you know there's Kauri DNA in there. And I had worked on Kauri as part of my PhD, and I knew Kauri, even when you extract DNA from leaves, does have a lot of inhibitors in it. And that would only get worse if you're using roots, and if you're using roots that are covered in soil, all of which increases inhibition. So we needed to figure out a way to make this process reliable. And the solution to that was Riley. Riley started a summer studentship with us in November and went about systematically taking the whole process apart, testing every component of it. So different treatments of the roots to the start, different chemistries for the extraction, different dilution series for the DNA extracts, different primer series, all of those things. She individually tested, compared, and figured out where the bottlenecks were and where the problems were. That was a lot of pipetting, and I can't really do it justice, the amount of different tests and tries that Riley was doing there. And I was at some stage worried she'd just go and I don't want to see any PCI anymore. But she figured it out. Essentially, with Riley's help, we ended up with a protocol that works reliably. And the way that would have to work is essentially this. You have your field team that does the collection and does the collection as they have done before. So as fast and as wide covering as before. They take the samples home, and at home, at the lab or in the office or wherever you want to set up the lab, starting on the next day, one part of the team will do the extractions. The extractions work like this. As it turns out, the crushing of the roots is actually counterproductive. It seems to increase the inhibition. Took ages to figure this one out, but a way to make this work is if instead of crushing the roots, you stick your roots into these little tubes of sand. They come with Kyogen soil extraction kit. They are designed to disrupt the soil in this first extraction step. Now we don't put soil in there, we put our roots in there together with a bit of extraction buffer, and then we vortex them for 15 minutes. That doesn't do much physically to the roots, but it seems to shave off layers and layers and leave the inhibitors back. And that seems to increase how well the extraction works. And if we then, after the extraction, also 1 in 10 dilute the extracts, then we get reliable amplification. And all of these components turn out to be important. So if we don't do this initial vortexing step, you can dilute as much as you like. It doesn't work reliably. If you don't dilute after the vortexing step, again, it doesn't work reliably. So it's quite sensitive to this whole process. But if you stick to this process, it works. So where are we at now? Essentially, what we have at the moment is a reliable PA detection system from root samples. It provides fast results. It can be done by the community. It can be done on site, but it can't really be done in the field. That just logistically doesn't work. So there has to be somewhere on site a lab that can be used where the extractions are done after the collection has been made. In our test, this was very, very reliable in picking up on PA in positive samples. It also never picked up any PA in negative samples. So all the tests that we can do were quite reliable and of course quite fast. So this whole process now with the shaking of the roots, which also speeds things up, now takes about, I'd like to say about 3 hours or so from start to knowing what's going on. Obviously, it needs ground truthing, so we need to test it against the current standard. We need to test it against alternative PCR-based approaches that, for example, Team Wellington has been working on, and we're trying to set that up at the moment. Now, obviously, that's all nice and good, and that was the objective for last year, somewhat achieved with some modifications, obviously. But then we also got ambitious about it, because we figured, wouldn't it be really cool if there was basically just like a COVID test that you can take to the field and you COVID test your Kauri for PA. And there's a way to do that. And here I have to credit Ben, who has been working on this now this year. Basically, there's not enough time to go into all of it, but basically it uses a CRISPR-based approach for detection. It is compatible with these lateral flow assays. And Ben has been working on developing this year. We've had some success in the lab. But again, not the topic for today. But this is where we're trying to go. Essentially, we would really like to make this a lot easier. There's a lot of steps that have to work before this works properly in the field. But just as an outlook, that's where we're trying to go. Simplification of DNA extraction would also be great. So any enzymatic work, and I know there are companies working on enzymatic tools for extracting DNA from roots, where you basically just put the roots into a mix and it dissolves DNA out. And that also would be a game changer in this context. So we've got something that works, but obviously there is a lot of room for improvement. With that, I'd like to acknowledge Te Runanga o Te Rarawa, Genomics Aotearoa, of course, for the funding, the Warawara Committee Kaitiaki, and Bioprotection Aotearoa also funded Riley's summer studentship. And thank you.
Thank you very much for that presentation, Michelle. It was really interesting to hear the ins and outs of the project and how you managed to overcome those problems that you faced. We've got a question in the chat that's popped up, so we'll probably flip between addressing questions in the chat and then some just general discussion or kōrero. So Marie says, sorry if the question is naive. Not a plant biologist, but would there be a way to test insects or arthropods feeding on trees, a bit like testing ticks for blood pathogens circulating in animals?
It's a really interesting question. What do the Phytophthora experts say? Is that would that organism be eaten by anything?
Can I answer that?
Yeah.
Is it okay? So when, I've been involved in I've been involved in Kauri Dieback research for quite some time now. And when it first showed up, they looked at all sorts of vectoring ways that Phytophthora could be transferred between trees and between forests and insects. Well, in theory, I suppose if they did manage to get into the xylem or in the soil and manage to ingest or absorb Phytophthora or spore or something like that, then compared to the other vectors that moved Phytophthora around, it wasn't something that warranted a lot of looking. So yeah, in theory, possibly, but it's not something that people would spend limited resources looking into.
Thanks for that, Amanda. We've got a question here from Jess. Do you know how far and how fast the spores can travel towards the bait? And Jess is also concerned for Carl and would like to know, is Carl okay? But main part of the question, how far and how fast can the spores travel towards the bait?
Yeah, again, I'd defer that to the people more knowledgeable about Phytophthora than me.
That sounds like it might be a question for Amanda.
No one's really looked down a capillary tube to sort of time how fast those spores make their way from being released to the source of the food, but they do eventually. But they have made some hydrological estimations how fast in the natural systems Phytophthora can move through soil pore water, and that's roughly about 3 meters a year.
Okay.
And as far as Carl goes, unfortunately, no, he's not well. I got sick last year and didn't get into the office for a certain amount of time, and I notified PhD student too late, and watering fell short, and he died of high water stress, unfortunately. Or he died of the flu, essentially, but it wasn't his flu, it was mine. Yeah, so yes, but he's still providing excellent samples. You can see him behind me. He's still helping out with training students. So yeah.
Very good. Can you see that next question, Michelle?
Yes, I see. Yeah. So do you use PA positive controls in your qPCR? No, I don't. There's a reason for that, although I don't know if it's a good reason. So I'm coming from an ancient DNA background, which means detecting small amounts of DNA in all sorts of circumstances, and that makes you quite nervous about cross-contamination from positive controls. Right? So I don't want to have this. Detecting false positive in a Kauri Dieback setting has huge implications. Forests could, be closed down. It has impacts for the community there, for people using the forest and so on. So the last thing you want to do is risk a cross-contamination from something that you have brought in. And for that reason, I'm leaving out the positive controls for the Kauri Dieback, and I'm only using the Kauri as an example. So what that tells us, it gives us an indication of the inhibition in the DNA extracts, but of course it doesn't tell us anything about whether it's PA-specific inhibition. We've tried the PA primers, tried them out quite extensively, and they seem to be very reliable. But yes, it's not as, it can't tell us about primer-specific inhibition.
Michelle, can you see that next question that's popped up in the chat from Ignacio? What measures are in place to ensure the sampling process doesn't accelerate the spread of the pathogen?
That might be a question for Hama, Takapari, or Aden.
That would be great if we could have one of our Kauri Dieback rangers answer that question.
Hi, kia ora. This is Aden. Just in general, a lot of cleaning. So with our equipment, we usually clean it with methylated spirits, but diluted, some of that, and a 30-70 mix of Dearden with water. Yeah, for the most part, we use that, and we just sanitize the living hell out of all of our equipment that sort of gets dirty, and then just making sure just everything's clean when we come back from the Ngāhere, from the forest. Another big thing is just not going to other forests in the gear that we use in our forest. So if we were to go in the forest and some other iwi rohe, we'd probably bring stuff that wouldn't really be too hard to clean, like just some red band boots, something that's not really going to be that hard to clean pretty much. But yeah, it can pretty much be simplified as we just sanitize everything with meth and Dearden and make sure we don't go to other places where we might be able to spread it. But kia ora.
Kia ora, thank you for that, Aden, and that's really comforting to hear that you've got those protective measures in place. Takapari, did you want to add anything to that? Or?
Yeah, just to add to what Moses was saying, we also, when we get to the Ngāhere, we have our boots in a separate bag, and when we do cross into the Ngāhere, that's when we put our boots on for that specific Ngāhere, and we do what Aden said, here we sanitize. Each Kauri hygiene zone that we do go into, we also sanitize all our equipment and our boots prior to entering in any Kauri hygiene zone, and while we're walking up. So every tree that we're soil sampling or every tree that we pass, we make sure that we're keeping ourselves contaminated free by sanitizing our boots, our equipment and our gear. And when we are working out of the Ngāhere, we're making sure that we're not putting anything on the ground. We hang things up on the trees just so that we're being mindful of not catching any disease that could be put on our bags if we put on the ground, or if we sit on the Ngāhere. So we're really mindful of what we do and where we work, and we're making sure that we keep those standards everywhere we go.
That's really fantastic to hear. Thanks, Takapari. Hama, did you have anything to add to what the boys have said, or they've pretty much covered it?
Yeah, no, they've pretty much covered it. Thanks, Tracy.
Cool. I'm not sure if you guys can see the comments, but we've got a comment that says "Ngā mihi mai oha, such worthwhile mahi." And good to hear about those protective measures that you're taking. Thank you. And we've got another question here, and it says, mindful of the potato blight and the chemical spray they used for this, has any chemical spray been found that the pathogen is sensitive to that you could spray or sprinkle the environment with?
I can talk a little bit to that. So there are a few commercially available chemicals not in New Zealand that have been tested, and they had reasonable success. But the trouble is they would only have success in a highly managed system like a nursery. You can't then go spray out basically fungicides. That's essentially what they are. Fungicides used in the cucumber industry against different sort of Phytophthoras and mycetes. And they can work. They did work. They had a pretty high success rate. I can't from memory, it was like 60%. But you would have to constantly spray, and so to take that and spray it in, primary growth forest, which has a whole lot of other species going on, it just wouldn't work, and for a whole bunch of reasons that you wouldn't know what all sorts of effects that I guess are the anticipated effects and toxicity and the other plants and other systems in there. So, or that you could use some of the nursery settings, you couldn't use it in our native forests. And at the moment, the only treatment they use is phosphate injections, where they inject phosphate into the main trunk of a tree, and the tree has to be a certain age. And there's a sort of a rough calculation about how much phosphate gets injected. But I haven't seen any published results that tell me that that's something that is worthwhile spending more time and resources on.
Thanks for that, Amanda. I just had a general question for the Kauri Dieback team. In terms of the work that you've done in this project, is that given you, or are you now better equipped in dealing with the Phytophthora threat? Has it given you a level of confidence that you didn't have before the project?
Oh, kia ora. So yeah, it does give us hope, and I guess to be able to look forward so that we can get the results back within a couple of days rather than waiting 6 to 8 weeks. So that will help us plan effectively to be able to plan in the Ngāhere where we can work and what measures we need to put in place to sort of control that. So to me, this is looking promising, and waiting. I can't wait till move forward and to start testing this.
That's great. I'm just gonna assume that as kaitiaki of Kauri that the project has really contributed to your kaitiaki role, which I think is great. You've talked to us about the sampling and ensuring that there's no increased chance of pathogen spread. Is there anything else that you've learned through this project with the sampling process any skills or techniques that you didn't have? I assume you were sampling before this project, but is there anything new there that you've learned in terms of sampling?
In terms of sampling, something new that I did learn is that it's a lengthy time to be able to do any testing on the soil sampling. You know, you mentioned that about how we go out and test. So we've been doing that for quite some time. But prior to this, we hadn't done any experiments in terms of soil testing. So this is quite new for us. And it just goes to show you, how much time that a person or scientist or people put into a project to be able to kickstart something to get it to where it needs to get to be able to move it forward. So yeah, there's a lot of hours in it to be able to get a positive or negative result from this DNA extraction.
Thanks for that, Takapari. We've got another question in the chat from Jean. What happens once you find an infected tree or an infected stand?
So just to basically answer that, we either tell the iwi hapu that there is a positive result, and then they'll come up with a plan in terms of protecting that area, whether it's to fence it off, whether it's to implement other safeties to tell the wider community not to go into that area because it's deemed to be unsafe, or to limit the cross-contamination. So those are some of the things that we look into in terms of there is a positive Kauri tree. And then just putting up signs so that people are aware where they're walking, and just bringing awareness to a lot of people who are in our community or iwi.
Some
Great.
hapu even put a full rahui on their ngāhere. We've got one up here in Hirakino. And that ngāhere has been, there's been a rahui for a few years now, so you can even, they can even do that as well. So that's another way of, they want to protect it.
Just for those in our audience that might not know what a rahui is, it's a prohibition. So it's usually used as I guess it's a conservation method, a culturally appropriate conservation method. Hama, where rahui have been placed, do you find that they're generally well respected?
Yeah, where it is at the moment, Hirakino, no one goes because that was part of the Te Aotearoa track. So the Te Aotearoa track's been, they've come up with another route.
Right.
Yeah, so people haven't been going there. So yeah, it's just a cultural prohibition to say like not to go in there. It's like restoring itself. Until then, you can't go in there until they make a decision to reopen it again. But yeah, we haven't done any testing in there since the rahui, so yeah, it's still in the rahui.
Thanks, Hama. We've got a couple more questions in the chat. Have you guys tested air mode for the eDNA for airborne spores of Phytophthora agathidicida? If we put air filters across the forests, would they provide any clue?
It's probably a twofold answer, which probably Amanda can speak to as well. So we haven't tried any air filters, and my understanding of the biology of Phytophthora is that it is very much soil-borne. So very likely it would be challenging or unlikely to detect it in the air. But then it's always easy to say unlikely and not having tested it. So not tested, unlikely to detect, but we don't know until we test, essentially.
Yeah, I mean, I'll just follow on with that. Michelle's right. It's sort of it's a two answer. It's a waterborne mold, basically. So it needs water to complete its life cycle. The only time that you might detect it in air is if you had dust from either a fallen tree that was infected, or it's a particularly dry forest season and vehicles or people have been, moving through the forest and generating dust, but that would be kind of short distances. And again, when you compare the gross movement of any infected material, dust would be a quite insignificant source.
Thank you. bioprotection using the root systems versus traditional methods, such as beta and morphological analysis, and considering the patchy distribution of PA in the soils, how much root sample do you collect? Do you sample around the tree or just one side?
So yeah, again, several answers. So first of all, in terms of the confidence compared to other methods, that is exactly what we're trying to set up testing at the moment. It's just working around everyone's schedules. Well, some might have already happened, but yeah, that's our very next step, and it's in process. Planning is in process. We just not and exactly we're going to manage it because it's putting a lot of things together. But that's the important next step before we can actually say, well, this is worth taking out and doing or not. The patchiness is another worry that we have, and which again, we need to test and don't know. The sampling is the same as it has always been. It will also continue to be the same as it's always been, which I think is eight soil samples per tree, if I'm not mistaken. But yeah, so it's a significant number of samples around each tree, and all of them go into the analysis separately. One question is, of course, the soil baiting obviously attracts the zoospores from a wider area and accumulates them. And how well the direct DNA extraction can resemble that, we don't know. This is what that test will hopefully tell us, whether it's more or less sensitive than traditional method.
I'm conscious of the time, which we're kind of out of, but we do have another question in the chat, and I was hoping to get a very quick roundup from each of the project team in terms of like a key insights or key learning from being part of the project, and then we will close with a karakia. So hopefully you've all got an extra few minutes on your Friday afternoon. But the next question is, have you considered that other forest plants might discharge chemicals via their roots or leaves that may inhibit or kill the pathogen? Was that yes?
Yeah, I was waiting for someone else to answer, but it wasn't forthcoming. I think that's probably for me, that one. Yes, it's a yes. Yes, we have tested other root exudates. Root exudates from plants often have antimicrobials as part of their natural defense system. There are a couple of native plants that are promising. One of them is Kānuka and Manuka. They have anti-Phytophthora properties. That's what we know. What we do with that, we're not sure in terms of developing a management tool going forward.
Thanks, Amanda. And while your mic is still on, could we just very quickly get some key that you've got from involvement in this project?
Yeah, so I mean, I think this is a great way to start a project that has, working with communities. There's a very obvious need. I mean, I've been working in this space for a very long time. Part of the bottleneck was always about testing and getting, information quickly to the people managing on the ground. We always had a bottleneck in terms of testing. There were only 2 kinds of labs, and of course you have to be molecular trained to detect that. And so getting this, working with the Kauri Dieback Rangers and getting Kauri Dieback Rangers familiar with what happens behind the scenes. So it creates a sense of trust, I guess, and sort of trusting the processes on both sides. And I think that's what's been great about this, that both sides kind of appreciate where each other are coming from.
Fantastic. Sticking with ladies first, Debbie, would you like to share a key learning or insight from the project?
Just more broadly, it's been really, really beneficial for Te Rarawa Rangers to have a research project that they can CoLab on. So much of the attention from universities and research institutes and CRIs is directed towards Waipoua Forest. And that's where Tane Mahuta is our largest kauri in New Zealand. So a lot of the opportunities go there. So really good for us to have this, for Te Rarawa and to have this opportunity. yeah, so big mihi to everyone involved. I think it was actually Fred from Biosense who kicked things off and brought Te Rarawa in. But yeah, that's been really beneficial.
Thanks, Debbie. Aden, would you like to share a key learning that you've got from the project?
Yeah. Kia ora. Well, actually, I've learned quite a lot. Yeah. Only 10 months ago, I was sort of in a different mahi, completely different to this, and being exposed to this whole sort of environment has been a real, well, I've learned a lot from it, really. Yeah, don't really know what else to say. I've learned all of it.
That's good to hear,
Obviously,
thanks.
yeah.
Yeah. Hama, have you got anything you'd like to share?
Kia ora. Yeah, I just want to thank Michelle, Amanda, and Peter for allowing us to be a part of this project. Yeah, it's a privilege and it's awesome to work alongside you know, the scientists. It is all new to us. It sounds like there's a lot involved behind the scenes. I mean, we're just the ground workers and we just pretty much do as we're told, and we'll get the roots for you because it's not too hard. But after watching what Michelle does and Amanda do with all the, you know, the filtering with all the systems, man, it's a pretty big job. So just want to thank you for allowing us to be a part of the project. And look forward to working with you and seeing what the end outcome is, if there is one. So, kia ora.
Yeah, for me, it's been awesome to work with the Kauri team because it's like this monthly reality check. Right? You know, when you're working in the lab, you kind of tend to get lost and you kind of forget what might work and might not work in the real world. And there's also, you know, different, like when you have your little science project somewhere, you want certain data, you can't get to it easily, then you make it more complex and then you get to it, or then you make it even more complicated and get to it, no matter what time it is or how long it takes to get it. But of course, that's not how it works in the real world. And so, you know, having that feedback from the team every once in a while, who were, I have to say, Takapari, Hama, and Aden were endlessly patient and polite about this. When I tried to present them with a method that takes half the day to just extract some DNA, and, you know, could have just told me to bugger off there. So having that feedback was really good, and I think that really helped putting something together that at least will work somehow.
Thanks, Michelle. Peter, we'll give the last word to you.
That's very kind of you. So, and I'm peripherally involved in this project. You can't speak on a Friday. And I had the privilege of going up and interacting with the team and seeing some of the Kauri forests and seeing all of the work that's done, which to avoid infection spreading between sites and things. I didn't get to see Warawara Forest, which, you know, seems like a really remarkable place. I'd really still love to go and visit. But also, it was a really good opportunity to see how actually we can bring lab skills into an Airbnb and build a lab in a kitchen and get some results. And that was all the hard work of the Kauri team and Michelle and Amanda, who did a fantastic job just getting through what is actually a really, really complicated procedure to get to the point where I think in that, when I was up there, I think we did the world's quickest detection of Phytophthora from sample to an actual positive. And that still was, you know, quite a long time. And since then, Michelle's got it further down. And I really look forward to the day when actually we can make that diagnosis standing next to the tree and make decisions on the ground really, really quickly, because I think that information and providing information quickly and effectively that can be acted on is a really key part of what genomics can bring. So yeah, it's been a privilege being involved, and it's been great to see the Kauri team in particular on the call today. So that's me.
Speaking of which, we'll just quickly see if Takapari is with us. And oh yes, he is.
I just want to tautoko what Aden and Hammers have mentioned, and to mihi atu Amanda, Michelle and Peter for being able to allow us to collab with them. It's exciting for us because it's meaningful to us that we're able to do our own soil sampling when that time comes, and it gives us more of a, for me, a sense of pride knowing that we're doing our own mahi rather it being sent away to someone else, and we're able to sort of give that knowledge back to our community, our hapu, our whānau, and to be able to share with them what we're doing in our ngāhere. So that's me, whānau. Kia ora.
Kia ora, Takapari, that was really heartening to hear. Sorry, whānau, we are well over time. Thank you to everyone that joined us today, and I'm sure you will all join me in congratulating the team for this research and the great collaborative effort. And I think that this project is an exemplar for mana whenua engagement, or for scientists engaging with mana whenua. And with that, Aden, could I please pass over to you to close us off with a karakia?
That's fine. Yeah. Mua pēnga karakia, tikana māku kia mihi a tātou kua tai mai ki tēnei hui, tātou o taua ao, pūtai ao, ngā tini, ngā mano o te ao pūtai ao, tēnā rā tātou katoa. Kia inoi tātou, kia tātou katoa, te ata whai o tō tātou arikia i Hukraiti, me te araho o te atua, me te whiwhinga taikitanga, ki te wairua tapu, ake, āmini. Kia ora.
Āmini. Ka ki te koutou.